Welcome to Biology Made Easy. In today's lesson, we are still discussing the type of differences in a species. We looked at heritable variation, genetic variation. You have discontinuous variation and continuous variation, which we are going to look at today. Welcome. So as we have said, in the species, no two individuals are the same, and variation is the differences. We have discontinuous variation in the species, and those are the characteristics or traits or phenotypes. They appear in the population in clear-cut forms with no intermediate forms. Clear-cut forms is also known as qualitative differences. Clear-cut distinguishable categories. You have such phenotypes. We look at sex differences. You have males or female. There are no intermediaries. You have tongue rolling, those that can roll their tongue, those that can roll their tongues. We have blood groups. We have people who can taste phenylethiocarbamide, people who can taste it. And so there are phenotypes that are very distinct, this male or female. On the other hand, there are also some phenotypes or characteristics that show complete gradation from one extreme to another, known as quantitative differences. For instance, height. In the species, you have height, very short people, medium, tall. You can't separate, so it's a complete gradation. That phenotype shows a complete gradation from one extreme to another. That's continuous variation. You have some phenotypes like that. And examples, you have intelligence, skin color, hair length, a lot of characteristics. Now, the graph is drawn like this. You have number of individuals, you have height, and most of the organisms always fall in the middle range for this variation. Then, in this one, discontinuous, you see, you can male, females, clear-cut forms. Now, the differences that cause discontinuous variation is brought about by major genes with no environment effects or very little environment effect. Major genes, you have one gene, tongue rolling, dominant allele plus its recessive allele. There are two genes. This is an allele for tongue rolling. This is an allele for non-tongue rolling. So these discontinuous variation is caused by major genes. Now, what about here? Continuous gradation in the species is caused by polygenes and environment. And polygenes are many genes, if you like, and each of the genes produces an effect. And the effect of all the genes will go to produce the phenotype. That is polygenes. And the effect of polygenes have environment affecting it so much. For instance, we say continuous variation, you have height, and height is brought about by polygenes. We'll look at the example soon. If you have all these genes, many genes coming together to produce one character, height, each of the genes produces an effect, and the accumulative effect will bring out the character. But the environment also has effect on producing the character. For instance, one can have gene for height, and yet if nutrition doesn't come in well at a proper time, the height that the person should have had will not go on. One will have a gene for heavy weight. Looking at the diet very well will make the person not have the weight. So polygenes. Now let's get back to here as we look at the comparison. So in discontinuous variation, clear-cut forms of individuals or phenotype, it shows Mendelian inheritance. And this shows polygenic inheritance. Mendelian inheritance, as we have looked at, you have a character like tongue rolling, is caused by two genes a dominant gene for rolling tongue, a recessive gene for non-rolling tongue. Mendelian inheritance, the allele of every single gene has a large effect on the phenotype. Each allele, so I have these two genes for tongue rolling. This allele has a large effect on the phenotype. Dominant gene T. Once a person has gene T, which is a dominant gene, the person will become a tongue roller. So in Mendelian inheritance, the allele of every single gene has a large effect on the phenotype. Then every different gene has different effects on the phenotype. For instance, this gene has an effect to become a tongue roller. This gene, this allele or gene, recessive, if it appears double, the person becomes a non-tongue roller. So every single gene has a large effect. These are tongue rollers, these are tongue rollers, these are non-tongue rollers. And every different gene has a different effect. This gene has a different effect, this gene has a different effect. Blood groups are also like that. Discontinuous variation. Now let's come here and look at continuous variation. It shows polygenic inheritance. And the allele of any gene has a small effect on the phenotype. For instance, let's take a phenotype like height of a man. As the man stands there, you take a feature like head. There can be genes, big A, small a, gene 1, gene A1, A2, A3. Head length, neck length, trunk length, lower limb length. All these have genes, and all these go together to produce the character height of a man. So if I take the neck length, these are the genes for neck length. Trunk length, these are the genes for trunk length. Lower limb length, these are the genes. These will produce the height of a man. And it's polygenes. Many genes will come together to produce the height of the man. And all the genes, their additive or cumulative effect will bring out the character. So, for instance, if I have an individual that has these genes, head length, it has big A, neck length, big N, trunk length, big T, lower limb length, big L. It will have a kind of a height. If I have another individual where head length, it has gene big A, but neck length, it has small n, trunk length, it has T1, lower limb length, it has L3, it will have a different height from the first one that had all these. So polygenic inheritance, as we have said, the allele of any gene has a small effect. These are the genes. Each one has a small effect. A number of different genes have the same effects on the phenotype height. So these different genes, they all have the same effect in producing height. The phenotype, that is the height, is produced by the additive or cumulative effect of many genes. So all the genes here will form the height. So no wonder you have a complete gradation from one extreme to another if the character is continuous variation. Yes, so we have looked at discontinuous variation and continuous variation. We want to take note that many characters or phenotypes that are discontinuous, like tongue rollers and non-tongue rollers, this will be the graph. But within the group of tongue rollers, you have the phenotype being continuous. So we will end up by having tongue rollers like this, continuous. For instance, you can see various types of tongue rollers. You can see various types of non-tongue rollers. And if measurements are made as to the lengths and the dimensions of various rollers and non-rollers, it will be a continuous variation. No wonder no two individuals of a species are the same. Another example is fingerprints. There are 8 major groups: loops, radial loops, ulnar loops, central pocket loop, double loop, hole, arches, or accidental. 8 major groups. But within each group is continuous. No two fingerprints are the same. So if I have loops, for instance, so I can have loops here, I can have radial loops, and each of them is discontinuous, but within each group is continuous. Even identical twins do not have the same fingerprint. Fingerprints of all human beings, past, present, are different. Every organism have a unique fingerprint. Fingerprints of all individuals start forming at week 12 in the womb, fetus, and the skin stretches in various ways acted upon by the environment in the womb to create unique fingerprints. No wonder no individual of any organism is the same, different. This is where we'll end today. Please drop your questions in the comments section. Kindly do subscribe, give us your likes, and look forward for our next lesson while we tackle why every individual of every species is different. Thank you. Goodbye.